First of all, I'd like to thank everybody for the opportunity to speak today. And when I originally was asked to talk about preserving diversity, I thought immediately of our work in terms of metagenomes. But then as I start putting this talk together, what I start to think more about was about other aspects of what preserving microbiome information means. So I'm going to present briefly on three different aspects of this. And before I start on that, I want to mention about why we think that preserving Australian human microbiome is important. And if we look in terms of population, Australia has one of the largest amounts of diversity in terms of where people were born. So in 2024, over 31% of people in Australia were born overseas. Another 22% of people were born from one parent that was born from overseas. So that's over 50% of the population. And this, in this list, you can see just the top 10 contributor countries for this diversity. And you can see how much diversity we have just in this setting. So there is an unique opportunity because of the diversity of the population to capture and preserve this diversity. So the first thing that we need to look in terms of preserving diversity is looking into preserving microbial species that may be different. And with that, I want to talk specifically about the work of two people that are leading the field in Australia, Professor Jane Tyson, that set up the Australian Human Microbiome Biobank. And this already has collected over 12,000 microbes, as well as the work by Associate Professor Sam Foster, who established protocols for established protocols for culturing what was previously believed to be unculturable microbes, and also co-founded the Biome Bank, which is a donor bank for fecal microbiota transplantation. The second aspect is then capturing and preserving information. And one of the key issues that we have in Australia is that we have very little representation in global studies. So in this study that was published in Cell this year, you can see that there was no samples from Australia in terms of the human microbiome. And this study, the preprint was just published last month. They identified 10 studies, but together they were less than 2,000 samples, and there was so much variability between the samples that I don't know if that is actually going to help to define what the Australian human microbiome looks like. There are also a lot of limitations in terms of the research that we're doing because of this lack of funding, especially for what would be classified as just blue sky research. There is also the need for multidisciplinary collaboration, and I think we're getting better at that, but we're not there yet. There's also a very diverse population in terms of where people are coming from, as I mentioned before, but also in terms of location, that Australia is a quite large country relative to a lot of European countries, for example. And this is still challenges in terms of the data analysis and our storing capacity of that data and how we're sharing and collaborating to analyze that data. And in terms of data analysis specifically, I do want to give a shout out to our group leader within our lab, Dr. Matthew Nelson, who during his time as the president of the Australasian Human Microbiome Research Network, set up a lot of training courses to upskill our community. So I want to take you back in time in the journey that I have taken in the past 10 years when I started working in this area. And if we go back to 2015 in Australia, we didn't really have human fecal samples available. We also didn't have the skills to analyze metagenome data, especially from a human microbiome and disease perspective. And we also didn't really share data, expertise, and skills. And more broadly at a global level, what I believe that this led to was a lot of studies that we couldn't really replicate, but also they were not biologically or medically relevant. So this led me to recruit my first cohort, and I'll tell you a little bit more about it, but also to look into establishing better guidelines and standards for the field, particularly starting with hypertension, that is our disease of interest, as well as just renal studies overall, and then contributing to some other guidelines and standards such as the STORMS checklist. So I'll tell you briefly about the first cohort that I recruited. And this is a very special cohort to me because I ended up having to be the person to go and recruit the participants myself and talk to them about the importance of their studies as well. And I learned a lot in the process. So this was the Victorian gut microbiome of the Vic Gut study. And we recruited participants into sites in the state of Victoria in Australia. So Melbourne, that's a metropolitan city, and Shepparton, that's regional Victoria. And this is important because we have a phenomenon in Australia that was coined the goat's cheese curtain by an economist, Bernard South, in 2015. And he coined this term based on the assumption that we have a lot of data showing that people that live in metropolitan areas such as Melbourne, they are more highly educated, and that means that they are more likely to consume things like goat's cheese relative to other areas in Australia. But what is really important about this is that it seems to create a bit of a divide in terms of not just the economy, but also it seems to create a divide in terms of cardiovascular health. So we know that people that live outside these major cities, they have higher rates of cardiovascular disease as a result. So we started this cohort looking into very detailed blood pressure measurements, looking into diet, the gut microbiome, and levels of short-chain fatty acids. And then after that, expanded the analysis to fecal metaproteomics and plasma metabolomics. And what was interesting is that we initially found no difference in the microbiome between people with normal and high blood pressure. But when we filtered and then looked into the specific mechanisms associated, we could see a very distinct, clear difference between people that had normal and high blood pressure, meaning that the mechanisms that the microbiome is involved in might be more important than necessarily just what microbes are there. We recently used the metaproteomics to look into this in more detail. And what was really fascinating about fecal metaproteomics is that we're able to detect over 1,000 human proteins in the samples. And these are proteins that we would not be able to detect in these participants without doing necessarily a biopsy. And after validating this across 2 cohorts in terms of fecal samples, we also looked into the ones that could then be excreted into the circulation and became plasma proteins in the UK Biobank, showing that indeed these proteins were able to predict long-term risk of having a cardiovascular event. And more recently, we also have been looking into integrating all the different levels of multiomics, so the metagenomes, metabolomics, and metaproteomics, to really understand some of the underlying mechanisms, because it's not just about what the microbes do, but then how they interact with our body to then determine human physiology. And we have been able then to recruit a series of other cohorts and also do clinical trials, including one with patients with heart failure, a clinical trial using an intervention using short-chain fatty acids, a study where we use the smart pill motility testing system to look into gastrointestinal pH and motility. Also, the HAPA trial that Dr. Matthias Nelson also leads in terms of gut permeability in vivo. And we are currently recruiting for the Grains BP clinical trial as well. And together, these are going to allow us to have more than 500 metagenome samples with very detailed phenotypes as well. So this is really important. So finally, I want to finish this talking about the preserving the diversity at the population level. And I want to highlight this study that I'm sure many people in the audience would be familiar with, that looking into the American Gut Project, they found that people that consume over 30 different types of plant-based foods per week, they had higher levels of short-chain fatty acid producers. And this is important because when we are talking about preserving diversity at the population level, it's not just about capturing what we have now and then preserving the species, but also looking forward into our communities. What do we need to do to help them preserve the microbial diversity and associated health as well? And this is in line with discussions that are even happening at the conversation. This was an article published just last week talking about the number of portions or servings of fruit and vegetables that we need to consume to have a lower rates of cancer and cardiovascular disease. So they mentioned this meta-analysis that looked into minimum 10 servings of fruit and vegetables a day, for example. And it's also in line with the American Heart Association approach for food is medicine, basically using food as an intervention for better clinical outcomes. And in line with that, our lab is particularly interested in dietary fiber, And through our interactions with the community, we have been particularly interested also in establishing guidelines, but also resources for our community that they can better understand how to increase their fiber intake, for example, that then can have a benefit in terms of cardiovascular health, but also in improving their microbiome. So what is next? I believe in Australia that we need a national coordination and funding for microbiome preservation and a better integration of how we look into microbiome in terms of clinical outcomes. But this is only possible if we are collaborating across different disciplines in terms of capturing and also then helping us to protect the microbiome diversity for our future generations. And perhaps it's time that we start talking about setting up an Australian human microbiome registry. Yeah, thank you.